students, in the last video we have studied the first feature of ideal vector, that is origin of replication, and we have started to study about the second feature, that is selectable marker. So selectable marker, it is used to select only the transformants and to remove non-transformants, permitting the growth of only transformants. Then the third one we have started, that is recognition site. So ideal plasmid or ideal vector should have one or few vector cloning site or recognition site to clone or to insert our gene of interest. Otherwise it will be difficult for the plasmid. So here we can understand one of the place sequence, it is BAM H1, and here the antibiotic, one of the antibiotic resistant gene is there, that is tetracycline. Here another one is there, ampicillin. So in the PBR322 there are two antibiotic resistant genes are there, that is tetracycline and ampicillin. Now what we are going to do here, by the help of BAM H1 we cleave or we remove this tetracycline and we are inserting our gene of interest. So here we are tetracycline removing and inserting our gene of interest. So this process you can say insertional inactivation. The process is called insertional inactivation. By the help of the word only you can understand, when we insert our gene of interest, tetracycline will be inactivated. So the process you can understand insertional inactivation. So now tetracycline is not there, our gene of interest is there. We have to find whether it is recombinants or non-recombinants. For that we have to take three culture plates we have to take. In one culture plate ampicillin, in the first culture plate ampicillin, and the second one contain tetracycline, another one containing ampicillin plus tetracycline, both will be there. So culture plates are there. Now we are putting our plasmid PBR322, this rDNA, into the culture plate. First this rDNA we are putting into the culture plate. Next again it is. So what happens here? In the first plate, the ampicillin is there in our rDNA, and here also it is there. So what happens here? It will grow. Our rDNA, it will grow in the first culture plate because ampicillin is there in the rDNA. In the second one, in the culture plate, tetracycline is there. So what happens? Tetracycline is removed already and our gene of interest is there. So what will happen? Whether it is growing or not growing, you can understand tetracycline is not there in the rDNA. So it cannot grow. It won't grow. So here you can understand it do not grow. And here contain both ampicillin and tetracycline. So here the recombinant also it will grow. So what we need, according to this observation, we need only the recombinants. So recombinants, it is not allowing, means it won't grow. By the help of that we can select our rDNA, our gene of interest, or you can say recombinants. So but what we can understand here, this is one of the difficult process because we have to take some culture plate and we have to observe whether it is growing or not growing. So some difficult process, it's a cumbersome process. Because of that, the another substrate is using here, chromogenic substrate is using, that is beta galactosidase. So by the help of beta galactosidase, we can identify whether it is recombinants or not recombinants. How it is possible? Listen here. What happens? We have to identify the particular sequence codes for beta galactosidase. And there we have to insert our gene of interest. In the beta galactosidase, we have to insert our gene of interest. So if that part, if it produce blue color, then we can understand non-recombinants. non-recombinants. If it produce blue color, then it is called non-recombinants. If it do not produce any color or colorless, then we can say recombinants. So by the help of beta galactosidase and by the help of color means blue color or colorless, by the help of that we can identify only the recombinants and non-recombinants. We can identify and we can select the recombinants and we can permit the growth of these recombinants. So these are the second feature, that is recognition site. And one more feature is that you can understand the next video. Please listen the video. Thank you, students.